Thank you very much, everyone, for being here and hear my talk about how to use mRNA cancer vaccine approaches to treat cancer. These are my disclosures. So 3 points that I wanted to make on my talk. Can we use the immune system to fight cancer? And how have we achieved that over the past few years? And how mRNA-based vaccines can help us in improving even more the use of immunotherapy in patients with cancer. The relationship between cancer and immunity is very complex and often paradoxical. On the left-hand side, you can see how much cancer growth begins and continues in a completely unchecked way, despite the immune system being present at the very early onset of cancer growth. So by the time a cancer is detectable, the immune system has already failed to control cancer growth, which is a true paradox, because a lot of cancers have a long latency phase. So there is a long time before the cancer is diagnosed. They're diagnosed on the background of inflammation. And as Robin said, cancers are different from our own healthy tissues because of different genetic characteristics, but also antigen characteristics. We have known since the early beginning of histopathology, thanks to Rudolf Virchow, 1863, that actually there is evidence of cancer having infiltration of lymphoreticular cells, of lymphocytes, which with better technologies we started to understand, particularly being cytotoxic T cells, so CD3, CD8 positive cells, which are the ones that somehow seem to be responsible for some form of immune control, or at least the relationship with the immune system in growing malignancies. The problem is that despite most tumors having evidence of an immune infiltrate, what happens in the context of tumors is very similar to what happens in the context of an infection that we can't clear. So if we get the flu or if we get an infection that we can clear, what tends to happen is that our immune system quickly develop an effector function, as you can see on the left-hand side. The antigen from the virus or the bacterium tends to trend downwards, and we develop memory against the specific antigens that are being exposed through our own adaptive immunity. But what tends to happen in chronic infections such as TB, hepatitis B, HIV, or indeed in cancer, is that we maintain a condition of saturation of antigens. So our immune system continues to get buckets and buckets of antigens, and therefore to avoid our own tissues being destroyed by an overactivation of our own immune system, what kicks in is a process called T cell exhaustion. So our T cells become a little bit more lazy. They tend to pick up certain markers such as PD-1 or CTLA-4, which are active, actionable drivers of anti-cancer immunity, which can be used therapeutically. And they basically instigate a state of dormancy of the immune function, which facilitates cancer to grow and is probably at the base of the paradox. We have been using the immune system to treat cancer way before we used chemotherapy, radiotherapy, or any other form of targeted therapy for cancer. A copy of the New York Times, 1908, where this crazy guy called William Coley started to inject bacterial toxin under the skin of patients with disfiguring facial tumors. As you can see, some of them had a clinical response, so the tumors became less ulcerated. And especially he noted that in those patients that develop a fever, the responses seem to be higher, which is consistent with the fact that through this particular use of antigenic stimulation from bacteria, he was able, perhaps through antigen mimicry, to induce an anti-tumor immune response. These were the origins, but the actual dawn of cancer immunotherapy comes from that Kaplan-Meier curve on the right-hand side. We as oncologists are obsessed with Kaplan-Meier curves, but this one was really, really revolutionary, because with the use of a drug which was rather obscure in 2010 called ipilimumab, a CTLA-4 inhibitor, basically up to 25% of patients with metastatic melanoma, a diagnosis that when I started working in medical oncology was inevitably fatal within 6 months, were actually able to be alive and well 32, 40, up to 56 months on treatment, which is really what gave credence to the concept of using our own immune system to treat cancer. And very quickly, going right into 2024, on the left-hand side, you have a cartoon that explains how many cancer indications are now being the subject of immunotherapy. Pretty much every single tumor under the sun has had some form of improvement by the use of immunotherapy. But what are the problems with immunotherapy? Well, you can see on the right-hand side that there are basically two key actionable drivers of anti-cancer immunity that I've translated into therapies that we can use today in the NHS for patients. This is PD-1 and CTLA-4. So these are a negative regulator of the immune response, particularly at the T-cell effector level, or the dendritic, the antigen-presenting cell. And basically, by inhibiting this pathway with monoclonal antibodies, we are able to get rid of that T-cell exhaustion that, as we have seen, is actually capable of reducing the chances of our immune system to recognize tumor. And we have moved on since 2010, and we're now using these therapies in combination with a number of other agents, including chemotherapy as well as targeted therapies, to try and broaden the reach of anti-cancer immunotherapy. However, there is a problem. Immunotherapy does not benefit everyone, and we've started to work out that there are a number of different problems in terms of instigated and effective T-cell response. So just by simply removing those negative regulators, we're not going to hit a lot of the sweet spots in terms of cancer therapy. Some patients develop primary resistance, so they never really achieve a response. Others respond for a while, and then they develop resistance later on, particularly after 6 months of exposure or longer. Plus, we also have other checkpoint inhibitor insensitive indications. Some colorectal cancer respond to immunotherapy, but the vast majority don't. Ovarian cancer doesn't respond, pancreas, as well as many others. And so we really need to further broaden the reach of immunotherapy by expanding the armamentarium of what we can use. And if we think about the mechanism of action of immune checkpoint inhibitors, which I have highlighted in red, these are not sufficient in terms of really breaking that tolerance, breaking that barrier between immune system and cancer, because they only really act on one specific aspect of cancer, of the cancer immune system interface, which is the immune effector component. You can see that there are a number of other characteristics: immune editing, antigen release and uptake, antigen processing and presentations, which are actually essential for the host immunity to develop immunity against cancer. And these are not tackled at all by the use of checkpoint inhibitors or chemotherapy or any of the strategies that we're using today in the clinic. And that's where therapeutic cancer vaccines kick in. So the idea is to provide instructions to our own immune system to make the characteristics of the immune response more targeted, more precise. And there are a few characteristics of successful therapeutic vaccination. First, the delivery of good quality antigen to dendritic cells, induce optimal dendritic cell activation, induction of strong and sustained CD4 and CD8 response, and then therefore infiltration into the tumor microenvironment, which is a very complex microenvironment made up of not only T cells, but also myeloid cells and many other negative regulators of an active cytotoxic T cell response, and also, of course, to maintain persistence and durability. So you can see that there are a lot of steps in that circle that need to be undertaken for a therapeutic vaccine to be successful. And perhaps it's not without surprise that we have been doing this for over 100 years since the first experiments by William Coley. However, I must say that the past 100 years, especially in cancer vaccinology, there have been, we can really label this experience as 100 years of skepticism, purely because a lot of the tumor antigens that were being tested with vaccinal approaches really didn't make it to the market. We're not really able to shrink the tumors effectively enough. And so a lot of the time when discussing about vaccinating patients for cancer, there has been really this strong sense of skepticism and due to lack of efficacy. This has really been due to the fact that a lot of the vaccines tried so far in cancer medicine were peptide vaccines. So where we basically take a protein and we inject it into patients. What's the problem there? Well, you have high specific and very safe compounds that can be used as peptide vaccines with very low risks, and you end up directly presenting the peptide to the MHC class 1 molecule. So this seems to be a really good way of doing it. But the manufacturing is very costly, and also you really need to have adjuvants to facilitate the dendritic cells to pick up the antigen, because otherwise you're just going to be left with a fragment of protein floating around with no immune response. Things have changed dramatically with the use of novel RNA and DNA technology, and particularly we have seen that with the use of DNA or RNA as vaccines, we have relatively lower cost platforms, and in particular, for DNA, we can think about a more stable sort of vehicle for that particular instruction to the immune system. However, with the risk of integrating into the genome and risk of having autoimmune reactions. RNA, definitely more rapid development, but the problem of fast degradation and actually the potential for an inflammatory reaction once the RNA vaccine is injected into the patient. And I think, as Robin alluded to, the key characteristics of RNA vaccine stems from our potential to really change the structure of endogenous RNAs into something that is clearly more stable. And as you can see, the green part is the part of interest to us, is really the target, the gene of interest. But there are a lot of other ancillary parts of a synthetic RNA, the cap, the polyadenosine tail, as well as many other modifications within the non-translated regions that facilitate that process of stability and make sure that these particular traits do become real drugs and not just nucleic acid floating around being degraded too quickly. We have already seen what happens once an mRNA, a synthetic mRNA, is injected. So the point is to engage with the antigen processing and facilitate epitope presentation, and then facilitate mature cytotoxic T cell to migrate directly into the tumor with a precise instruction. And we have seen what are the desirable properties of an optimal mRNA platform. But my point, using that IKEA kind of sort of instruction booklet to build up furniture, is really what is the target? What are we going to instruct our immune system to build once we inject an mRNA vaccine? And this is really the key point. Central to anti-cancer immunity is the presentation of the antigen, really. And so the entirety of cancer immunotherapy approaches that we've been using so far heavily rely on adaptive immunity. And we can think of mRNA technology purely as a toolbox that still require hypothesis-driven clinical research and knowledge of cancer tolerogenesis to make sure that we use antigens that are really key to either as off-the-shelf or personalized approaches to improve and increase the number of drugs that we have available for our own patients. And so there are many different antigens that tumor can express. Some of them are called tumor-associated antigens because they're not, strictly speaking, proteins that are only present in tumors. They're also present at low levels within healthy tissues, but tend to be overexpressed in cancers. These are important targets, but they have got a problem. They are subject to central tolerance. So our own body actively eliminates those T cell clones that respond to those cells because those proteins, those antigens, are identified as self. Then we have other proteins that only really get expressed as normal, structurally normal proteins if a cancer is present. So these are often oncofetal antigens. So often in the process of deregulation of cancer, we have antigens that get expressed, and there were dormant proteins normally present in the process of embryogenesis. A lot of cancer are virally driven, and so there are viral proteins that can act as antigens. And actually, as Robin said, because cancer is a disease of our genes by a large extent, any specific mutation that leads to a structurally different protein might actually be identified as a tumor antigen. And so there are many different characteristics that make a good antigen such. The more foreign the structure of the antigen is, or the epitope per se, the more likely it is to initiate a T cell response. Another important characteristic is the clonal distribution of antigens. So unfortunately, cancer is a heterogeneous disease. So not every single cell is equal to another. And so the idea of having clonal antigens, so antigens that are widespread and distributed across most of the tumor subpopulations, is probably the type of antigen that we want to go for. Some mutations are driver, some mutations are really linked to the mechanism of growth of cancer. Other are more sort of passenger mutations, and they may not be relevant to cancer growth. So we really want to hopefully focus on the ones that are more functionally relevant. We need to make sure that those antigens are adequately presented and they can actually bind very well to both the MHC class 1 or 2 as well as the T cell receptor. These are the interactions that facilitate the immune system to be active. We have already seen how we can try and identify the right antigen, and hopefully how to make this also a patient-driven and a patient-specific process. So there are a lot of computerized models that can help us predicting on the basis of the characteristics at the genomic level of the tumor, what are the most likely genomic alterations that can lead to good antigens. Sometimes this can be done also at an individual patient level. We have made incredible advancements in terms of technologies such as immunopeptidomics, where we basically mulch an entire piece of tumor. We try and dilute the HLA and proteins, and we try and evaluate what are the actual antigens present in the tumor that are being actively recognized using mass spectrometry. Or we can use more T-cell based assays by identifying either tumor infiltrating lymphocytes or peripheral lymphocytes from patients with cancer, and try and incubate with antigens and see which ones are actually eliciting a response. And the combination of all these approaches can actually identify shared or personalized antigens, as we've seen before. I've just tried to sort of chuck a few words to try and understand what are the clinical and immunological considerations to make in terms of choosing between a shared versus a personalized mRNA vaccine approach. And I think these are all important points. So how scalable the technologies might be, the turnaround time. Very often, as we said, patients with cancer need treatment quite quickly. So having an off-the-shelf approach is useful because it doesn't really require prior knowledge of the individual patient characteristics. However, on the other hand, with personalized approaches, we can count on more precise mechanisms of action that are truly relevant to that particular tumor. There are other aspects such as resistance or breadth of applicability that do require testing. As Robin said, this is not a particular type of approach that we can use in the NHS outside of clinical trials at the moment. And so we've already discussed how a personalized vaccine can really take place. So you can see that there are many different steps that are required, isolation of patient's tumor. So this is something that at a patient level is very important. We require a lot of tissue sometimes to generate a signature using exome sequencing or RNA sequencing. And very often patients have to undergo fresh biopsies to ensure that we have an up-to-date immune profile or mutational profile of the tumor. And then with in silico models, we can try and match the contexture of the tumor with the characteristics of the antigens that we want to explore. Problem is that the turnaround time for some of these technologies in clinical trials can be very long, up to 12 to 16 weeks. There is a lot of risk of failure at each step. And there is also the requirement for immune monitoring. So once we have an antigen, we need to prove in the individual patient through repeated blood tests that there is actual engagement of the immune system with that particular antigen. There have been a couple of really important success stories in cancer immunotherapy, and I wanted to focus particularly on a couple. This first one is Keynote 942. So this is a program that has been recently published in The Lancet. This is a form of personalized vaccine where, as you can see on the top left-hand side, patients will undergo exactly the same workflow that we said before. So sequencing of DNA and RNA of the tumor, HLA typing through a peripheral blood test. And as you can see on the workflow before, on the bottom left, in this particular clinical trial, 70% of the patients that were screened actually ended up being able to be randomized to the study. So even the failure rate of all this particular technology was in the region of 30%, and patients were basically randomized to receive mRNA 4157, which is a cocktail of up to 34 neoantigens into the same vaccine. As you can see on the top right, these patients that were treated had definitely evidence of an immune response. So you can see the red bars are basically a trebling or more of the immune response against the specific epitopes that were put in that particular cocktail. But what is more important to me as a practicing oncologist is, again, that Kaplan-Meier curve, which shows how patients treated with this particular vaccine, these were patients with melanoma fully cured, that were receiving the mRNA vaccine together with the PD-1 inhibitor pembrolizumab, while those patients were actually characterized by the improvement in the recurrence-free survival, which means a delay and an obliteration of the risk of recurrence in people that were fully treated for the cancer, at the price of very low toxicity. So overall, these vaccines are very tolerable in patients and do not impose at all in terms of their quality of life. Another perhaps more important milestone is the publication on Nature last year of this other personalized vaccine called autogene cevumeran. So this particular trial looked at very hard to treat patients with pancreatic adenocarcinoma. These are patients that are refractory to any form of immunotherapy. And as you can see, they were treated surgically And following this, they received the standard post-surgical chemotherapy. But before that, they were vaccinated with at least 8 priming doses of this personalized vaccine that was, again, derived exactly the same way by taking a tiny bit of tumor tissue as well as peripheral blood, so that the perfect and selected neoantigens for that individual patient could be derived. And before the priming doses of the vaccine, a single dose of atezolizumab, a PD-L1 inhibitor aimed at boosting the T-cell responses, was given to these patients. Well, the message is quite clear. On the top right corner, you can see that a lot of these patients, especially the responding patients, actually had evidence of immunogenicity. So in the peripheral blood, there was clear evidence of the patient's immune system picking up those neoantigens and starting to actively respond to those, which translated beautifully, again, in these other Kaplan-Meier curves, in a clear benefit in terms of recurrence-free survival. Obviously, cost, expertise, turnaround time, and the fact that only patients resected with pancreatic cancer, which are the minority, unfortunately, could potentially benefit from this approach, still limits the validity, the external validity of this approach. But this is something that I'm not failing to define as a breakthrough in a disease that is very, very hard to treat, and where the survival is very often limited to less than 1 year. So there are a number of leftover challenges in this field. So we know that mRNA vaccines can actually tackle a number of issues when it comes to resistance to immunotherapy. So for instance, impair antigen encounter, impaired effector function. But actually, there are a few things that cancer vaccines can't do. And so they really need good help from other modalities that we tend to use in anti-cancer therapy. So mechanisms of resistance to cancer vaccines might have to do with the fact that, unfortunately, the tumor microenvironment is spontaneously geared towards immune suppression. And it's not really just about T cells. There are myeloid-derived suppressor cells, M2 macrophages, NK cells, a lot of other actors that are not really being hit by a vaccinal immunity that we really need to think about tackling to further broaden the reach of this approach. There are also adaptive changes in T cell engagement. So very often through the process of neoplastic progression, tumor cells lose tumor antigens. They lose the mechanism of HLA and MHC class 1 presentation. And so they tend to escape the capacity of sort of being targeted by vaccines potentially. And there is the last aspect, that if you have ever treated a tumor, you do realize that no matter what you throw at them, there is this idea of cancer always coming back, of always developing processes of clonal selection and immunoediting. And this is certainly true. There is evidence of immune escape from vaccines too, which might suggest that to really make the best use of mRNA vaccines, we can't really think of them as single agent therapy, but try and combine treatment with non-overlapping mechanisms of action. So I hope I managed to convince you that it's truly exciting to be working with mRNA cancer vaccines, because they do really have the capacity to increase the chances of us using cancer immunotherapy more precisely and with higher potency. The debate is still out in terms of whether personalized versus shared antigen approaches are the best way forward. But I must say that if we go for personalized approaches, we might actually see dramatic changes in the way we think about anti-cancer therapy in our NHS resources. And so we really need to think about that too. We need to think about therapeutic resistance to vaccines. The fact that we're now starting to use them in clinical trials puts a lot of emphasis on trying to ensure that we don't waste an opportunity to understand why some of these patients actually fail to mount an immune response. And the last point I think is particularly crucial. We really need to maintain patient and public involvement very high. There's been a lot of vaccine hesitancy during the COVID pandemic, and I can't think about a bigger threat about us putting a lot of investment into cancer vaccinology, and then patients actually feeling scared or not wanting this particular treatment because of them, not really understanding the basis of what we're doing for them. And with that, I thank you very much for your attention.